Both RNAi and CRISPR are used for gene expression. They both have the ability to silence genes through different mechanisms, sometimes referred to as knockdown or knocked out. These mechanisms can be applied for medical advances such as gene therapy and new ways to treat diseases. If a bacteria or virus invades a cell, a double-stranded RNA is cleaved by an enzyme called dicer into fragments. These fragments are called small interference RNA, which is abbreviated as siRNA. Each siRNA contains about 20 base pairs designed to match a complementary target messenger RNA. The siRNAs are then attached to a protein complex called RNA-induced slicing complex, aka RISC. RISC matches the siRNA to the target mRNA and cuts through the gene, degrading mRNA and inhibiting transcription. If the dsRNA originates from a pre-miRNA in the genome, then dicer cleaves dsRNA into microRNA fragments. These fragments bind to a RISC protein to pair bases and match with mRNA. This process inhibits translation and therefore silences the gene. Both pathways lead to gene silencing or knockdown, which means gene expression is reduced, not completely eliminated. CRISPR has been used as more of a gene editing tool and basically acts as a bacterial immune system. First used for viral infections, scientists recently discovered how to use it for gene editing. The process is similar to a copy and paste mechanism and can alter genes like fixing a typo. It uses short snippets of repetitive DNA sequences called clustered regularly interspaced short palindromic repeats, aka CRISPR, and a CRISPR-associated protein called Cas used to cut through DNA. This process designs a guide RNA to match a target gene. The gRNA is then attached to a Cas9 protein that travels to the target gene where DNA is cut. The cell tries to repair the broken gene, but is prone to mistakes and often leads to mutation, turning off gene expression. The fixing process starts by making a template strand of the ideal DNA sequence. This strand is added to the cell, so the broken DNA strand can use it as a model to repair itself. This can repair defective genes or insert a completely new one. Some refer to this process as a knockout, which means there is no detectable gene expression. I would choose a human cancer cell as my eukaryotic cell and CRISPR as my method because I think it has incredible potential and it's amazing to see humans creating and mastering ways to manipulate genes. There are so many potential cures for various diseases through CRISPR. This could be a huge jump in cancer research, which has exploded recently due to various unpredicted causes. I would also use CRISPR because it is fairly new and not enough research has been conducted. This could be a good experiment to see if any problems occur in the future generations or just to watch the effects. There are many ethical problems that come with CRISPR, but I think it's worth putting the effort towards it and it deserves more attention.